Hello, this is Dr. D, and today we are going to talk about adaptive immunity. What did we say was adaptive immunity? Adaptive immunity is a specific resistance to a particular pathogen, toxin, or a metabolic byproduct of a pathogen. Remember, it is very, very specific. Not any metabolic product, not any toxin, not any pathogen, but a specific pathogen, a specific toxin, a specific metabolic byproduct. All right. So how does it recognize this? It recognizes this with an antigen. What's an antigen? An antigen is a non-self-specific defining molecule to which immune system responds. It has to be a specific molecule. It could be a protein, it could be a polysaccharide, it could be a glycoprotein, it could be a glycolipid. It has to be a well-defined molecule which is non-self. That means it doesn't belong to that particular body. All right. So here, self versus non-self, immune system has to learn about the self molecules before birth. And the learning process, this is why that education of the T and B cells, education means maturity of the T and B cells is important. So here, the T and B cells, what they do is they generate an immune response against, antigens. If they are not educated, if they're not taught, they are going to attack the body against, it's called autoimmune disorders. All right. So here, let's see. There are the two types of lymphocytes. We talked about the T cells and the B cells. What? Both of them originate in the red bone marrow. The difference is the T cells, that they will migrate from the red bone marrow once they're formed to the thymus, where under the influence of the hormone thymosin, they will mature. They are educated and understand the difference between what? Self and non-self. So obviously you see that there is more of the T cells and less of the B cells. And I will ask you why later on. Okay, so keep this in mind. So the 70 to 80% of the lymphocytes are the T cells. Only 20 to 30% of the lymphocytes are the B cells. The B cells remain developed in the red bone marrow. All right, and later on they will, once they mature, they go to the lymphatic organs. But for maturity, they do not have to migrate to any other organ. All right. So again, the T lymphocyte, the type of immunity they provide is the cell-mediated immunity. Why? Because they interact directly with the pathogen or the foreign body that is displaying the antigen. All right. The B lymphocyte, the type of immunity they provide is called humoral immunity. All right. So here this is stimulated by the antigens, and the B lymphocytes, they do not interact directly, but what they do is they produce antibodies. So they themselves don't directly interact with the cell. They don't go after this foreign body. They will produce soldiers. So these will produce antibodies, which are the proteins, immunoglobulins, that will bind to the antigen and destroy that pathogen displaying that antigen, that unique molecule. All right. So here, when the lymphocyte encounters, they become what is called as activated. All right. Let's see the types of lymphocytes. Now, the first type of is the cytotoxic. The cytotoxic, they will recognize the antigen. Antigen, remember, is a unique non-self molecule on the cancer cell or the virus-infected cell. They become activated and they will kill the foreign body. This is cytotoxic. There are the helper T cells. I'll talk about the helper T cells just once, but basically after activation, they will produce substances that will stimulate a B cell. Give me a minute, we'll talk about that. Then there's a memory T cell. Memory T cell is also produced after stimulation, and what they do is that they're kept in reserve. So future infections, what happens is that memory T cell will launch an immediate response, will directly attack that, will recognize that foreign body, that foreign antigen presenting body, and will destroy it. All right, this is a memory T cell. Regulatory T cell are the ones that will keep all the T cells in account. They're like the class monitor. They will make sure that the T cells are only attacking non-self molecules. They are not attacking self. All right. If they attack self, if these regulatory T cells, suppose they are non-functional, then what happens is some T cells become rogue and they will attack their own body, and that's how you end up with autoimmune disorders. A B cell will recognize an antigen, become activated, produce antibodies that will now attack the antigen presenting foreign body. All right. So let's look at the activation of first, how a T cell is activated. All right. This is the macrophage. So what a macrophage does is the macrophage first is going to engulf and swallow a foreign body. All right. Once it engulfs and swallows and destroys a foreign body because of the lysosome, then it will digest it and will separate the self from the non-self molecule. All right. The self molecules are not of interest, but the non-self unique molecules that was found in the foreign body, these are presented on the macrophage at certain sites. And these sites are called the MHC. What's that? Major histocompatibility complex. All right. So here what happens is that these macrophages will present this foreign molecule, and now this foreign molecule is called epitope. All right. So this epitope is presented on these MHCs, on this major histocompatibility complex. Or what? What is this large cell? This large cell is nothing but a macrophage. All right. Now what happens? Now, watch what I said. Now a helper T cell comes along, and a helper T cell now will bind to that epitope. Remember, helper T cell will bind because of a particular hinge, a binding site, and that binding site of a helper T cell is called CD4 or CD8. Let me see if I've said this out here. Okay. Anyway, so here a helper T cell, notice, is binding to the epitope. Okay, so here's the macrophage. The macrophage is presenting the epitope on what? On what is the site called? What are these sites called? MHC. So here the helper T cell will bind this via a CD4 or a CD8. Got it? So this complex is necessary. Helper T cell, CD4, CD8, epitope, MHC, on what? Macrophage. Once this helper T cell is attached, it'll wait. What will it wait for? Meanwhile, a B cell will also bind to the foreign body. Via what? Via these binding sites. All right. So here what happens is it will bind to a particular antigen of that foreign body. So when only it binds to a foreign body, so it has to bind to the same antigen as the helper T cell. They have to match. So if the helper T cell is bound to that particular antigen and a B cell is bound to the same antigen, then they will start communicating. A helper T cell will now release certain chemicals called cytokines. These cytokines will activate this bound B cell. Will it activate these cytokines? Will it activate other B cells which are unbound or bound to a different antigen? Answer is no. It will activate only those B cells which are bound to the same matching antigen. And when this happens, then what happens is that this B cell will become activated. And once this B cell becomes activated, it forms what is called the plasma cell. And these plasma cells are going to produce antibodies. These antibodies are designed to bind, to destroy that particular antigen, not any other antigen, but that particular antigen to which the B cell was bound and the helper T cell was bound. They have to match. Remember what I said. Some B cells will form memory cells. So remember, memory cells are formed both by T cells and with B cells. All right, keep this in mind. This is going to be important. So that later on, if there is a future infection, you don't have to go through this entire saga. Right away, the memory B cell will produce what? Antibodies. All right. So the shelf life of these B and T cells is number of years. All right, is about 20-25 years. So, okay, let's look at this. So here, this is called the polyclonal response. So pathogens, when they are going to, remember, pathogen will have many antigens, right? So remember, the different types of T and B cells will attack. And so what happens is that is why it will help to destroy the foreign body. It's just not like one antigen only will activate one T cell and B cell. No, there are many antigens, and different groups of T and B cells will attack it in as a team. All right. So here's an antibody. An antibody looks like this. So there is there's a heavy, it's protein in nature. So there are chains, right? So there are two long chains and there are two short chains. The inner chains are called heavy chains. The outer chains are called light chains. And these chains are connected. They are joined. All right. So here there is a constant region and then there's a variable region. Now this variable region is the one that varies from antibody to antibody. And all of these are joined, these four chains, 1, 2, 3, 4. What are these chains of? Amino acids. These are protein chains, okay? So these chains are bound by disulfide bonds. So there are different types, which is again very important. There is the IgG. IgG is the one which is the maximum in amount. And here what happens is these activate the complement system, promote phagocytosis, and they can cross over the placenta. Keep this in mind. They're also important for the Rh reaction, right? IgM is the one that activates the complement system, and they're responsible for the ABO blood type. All right. The IgA is the one that's secreted in the what? In the saliva, in the tears, as well as in the first milk, the colostrum. IgE is the least common of all. And IgD is the one that binds to the B cell and acts as antigen binding site. We'll complete the rest of the presentation and the next presentation, all right? So I thank you for listening to me. There's a part 2 too.